Let's take a closer look at enzymes in this video. First question, what is an enzyme? Well, let's think about a chemical reaction that takes place inside a cell. Let's imagine that we just drank a big glass of milk. And let's also imagine that we're lactose tolerant because otherwise, ouch, and we can actually digest it. Lactose tolerance, by the way, is actually much less common than lactose intolerance in humans. Okay, so lactose tolerant people are actually the weirdos. One of the components of milk is lactose. You're probably familiar with this as the main sugar in milk. Lactose is a disaccharide, and it can be broken down into two simpler sugars, glucose and galactose, both monosaccharides. And this reaction is a spontaneous reaction, meaning that it could occur on its own, but there's no guarantee that it will, and there's also no guarantee as to when that might happen. But if we drink milk, we need that to happen now so that we can digest the substance and not have a stomach ache. So what we're going to do is use an enzyme to catalyze this reaction. Catalyze means to accelerate or to speed up the reaction for it to happen, to enable it to happen. And enzymes are molecules that are biological catalysts. So they're biomolecules that speed up, or in other words, enable reactions. Enzymes are almost always proteins. We've learned about proteins, and you know now that they're long chains of amino acids that are joined together and then folded up into a structure that can do something. There are lots of different types of proteins, and enzymes are just one of those types, but they're super important. So for this reaction, we're going to use an enzyme called lactase. This enables the breaking of lactose into its two subunits. Metabolism absolutely depends upon enzymes. At every step, we need an enzyme to help the reaction proceed. For example, let's take glycolysis. This is the first part of cellular respiration in which we break down glucose in order to produce ATP. Glycolysis has 10 steps, and each one is catalyzed by a different enzyme. How do enzymes work? Well, they rely on the special superpower of proteins that we've discussed before. The ability to bind to other molecules in a very specific way, and often a very reversible way. Enzymes bind to reactants of chemical reactions and make it easier to do things like break chemical bonds or make chemical bonds. Here's a picture of the enzyme glucosidase. It's similar to lactase in that it takes a disaccharide, in this case maltose, and breaks it into two monosaccharides, in this case, two molecules of glucose. Maltose binds to the protein by interacting with amino acid side chains at a certain place on the enzyme, which is called the active site, and it's shown here in red. We call maltose the substrate of this enzyme. A substrate is a molecule that binds to the enzyme and upon which the enzyme acts. Binding between enzyme and substrate takes place via chemical bonds, the chemical bonds that we've already learned about, such as, hydrogen bonds. Enzymes are also really specific. They tend to only bind one specific substrate molecule, or maybe just a couple of molecules that are really chemically similar. Let's also take a second to remember that proteins are really dynamic. Like all molecules in the cell, they're actually moving all the time and slightly changing shape all the time as they flex around their chemical bonds. When the substrate binds to the enzyme, we have what's called an induced fit, meaning that the enzyme, and also the substrate, by the way, slightly change shape. This helps the reaction take place. It enables a closer binding relationship. Once we have an enzyme-substrate complex like this, then the reaction can proceed. Enzymes can catalyze reactions in different ways depending on what type of reaction it is. They can do things like twist substrate molecules to make it easier to break those covalent bonds, for example. Or they can provide a little microenvironment with a higher or lower pH than the surrounding area that makes it easier for the reaction to proceed. Sometimes they even make covalent bonds temporarily with this substrate, but however they accomplish the reaction they are catalyzing, once the reaction is done, the enzyme will release the product and exit the reaction fundamentally unchanged, meaning it can then go on and catalyze the same type of reaction with another identical substrate molecule. So, to summarize, we have an enzyme which binds a substrate via induced fit. The reaction takes place and the substrate is converted into the product or products of the reaction, which then dissociate from the enzyme. The enzyme itself is not changed by the reaction and it can go on and repeat the whole process. When we write chemical reactions, we write enzymes next to the arrow, often over the arrow, for just this reason, because they're participating in the reaction, but they're not actually being changed by this reaction. Okay, so that's our brief overview of enzymes. Next time we'll talk more about how they're actually regulated in a cell.